My name is Astrid Schnetzer. I'm an associate professor in the Department of Marine, Earth, and Atmospheric Sciences at North Carolina State University. And I have the pleasure to talk to you about a study on harmful algal blooms and associated algal toxins in Falls Lake, supported through the NC Policy Collaboratory, where we work closely with NC's Department of Environmental Quality. As you might be aware of, harmful algal blooms, or HAPs, are increasing in frequency, magnitude, and geographic extent. And if these algal blooms occur, they can have consequences for drinking water resources, recreational use, excess biomass can lead to the deterioration of water quality. And if the algae that are blooming are also toxic, there are additional concerns in regard to the risk of exposure to these toxins. Just as a reminder, algae are really important for any kind of marine system or freshwater system. They're at the base of the food web, and these primary producers support all the life in these systems. However, with increasing eutrophication and associated with some climate drivers, we might see harmful algae more pressing concern in the future. Agencies and others that are bestowed with water quality research typically look for certain things that can give some indication on HAP dynamics in a system. And some of the metrics that are often used are chlorophyll concentration or cell counts. Now that toxin measurements are becoming a little bit easier to do, we can also see more of those toxin measurements show up. The World Health Organization has created recommendations or categories for these metrics that can tell you whether there is a low, moderate, or high exposure risk to cyanotoxins, in this case, microcystins, which is the most common group of cyanobacterial toxins in these systems and the best studied. Based on these metrics. So in other words, you see right down here, the chlorophyll concentrations in the upper and lower Falls Lake as an average over several stations with the standard deviations over our project period. And if you overlaid based on these chlorophyll concentrations and based on the WHO categories, we would find ourselves within the category of moderate exposure risk to microcystin based on chlorophyll. Microcystin is, however, not the only toxin that is commonly cited in these systems. We also have cylindrospomopsin, enatoxin, saxitoxin, and BMAA, and all of these can have short or long-term health effects. Our project was sampling for all of these toxins. So we were going out with the Department of Environmental Quality and sampling and collecting samples at all of these 11 stations that you see here. And we were analyzing samples for chlorophyll, particulate, and dissolved toxins. So the part of the toxin that is still inside the algae versus the dissolved fraction in the surrounding water. And then on 4 of these locations, we also deployed in situ tracking devices. I will explain what they are in the next slide. To date, we have finished 18 monthly surveys. We only missed January and February of this year due to COVID restrictions. A little bit more about the methods. So we sample using bottles and finite volume at the particular time when we go out. And then we look at both dissolved and cellular fractions. The reasons why we distinguish between those is because they will take different pathways or create different pathways to toxin exposure that are relevant. The dissolved fraction is more relevant for drinking water concerns, while the cellular toxin can be consumed and passed on through the food web. There's also a third path, aerosolization of these toxins, that have more becoming a focal point lately. So when you do this grab sampling approaches, the problem is that if you go out biweekly or monthly, that you might actually miss the peak of a bloom. These blooms move around spatially and they're very ephemeral, so they can create and come and go and dissipate within days. So to also get a better idea of what is going on in the environment, we complemented our sampling approach by using solid phase adsorption toxin tracking, or SPAT. These are little resin bags that are put in the water, and they accumulate dissolved toxins over time and give you a time integrative signal that might also allow you to get at toxin concentrations that are typically missed because they're too low when you do grab sampling. The only downside on this is that you get only the dissolved fraction. And also what you get as a result is a semi-quantitative measurement, nanograms of toxin per gram resin per day. So you don't know if you have them out for 2 weeks or 4 weeks, was that accumulated within 2 or 3 days during the peak of a bloom or continually accumulated over the entire deployment period. So there's no way of directly linking to regulatory thresholds as you can with grab sampling. So after this quick intro to the methods, here's some of the preliminary results. We have at this point analyzed 880 samples. We've categorized them in particular dissolved and spat samples, and you can see them here listed for the 5 different toxins. As a very coarse overview, we find both microcystin and anatoxin commonly, cylindrospomopsin only so far in the dissolved phase, which might be an indication that we're missing the peak of the event when most of the toxin is still contained within cells. We have not found saxitoxin at all, which is good news, and we're finding BMAA very sporadically. So toxins are present. What are the concentrations? So what you find here in this graph is the average concentrations over the entire lake with the standard deviation on the primary y-axis up to 1 micrograms per liter. We see microcystin, cylindrospomopsin, and anatoxin, and then BMAA, the square symbols, BMAA is on the secondary y-axis, typically in slightly higher concentrations. Two of the main takeaways here is that there's multiple toxins present at the same time at the same locations. The other thing that is you should be taken away from here is that there's the potential to chronic exposure year-round. However, the concentrations per se do not exceed 1 micrograms or less than that. And if we look at the drinking water thresholds or the recreational thresholds through the EPA, you can see that we're sort of like in the lower zone here, and there is no immediate concern. However, keep in mind, these are grab sampling monthly data, so we do not entirely know whether there is peaks that we're missing. So if we take a step back and we look at where we found ourselves just using chlorophyll as a metric, we can now compare using microcystin directly as a metric, and we find that we drop into the low exposure risk category that is between 0.1 and 10 micrograms per liter. We're also starting to decipher some of the spatio-temporal patterns here shown for microcystin, which is really interesting. We see here the stations from the most northern to the most southern side over the time period. The blue squares show you when we did not detect the toxin or microcystin, it fell below the detection range of the method. And then you see the colored scale all the way to 0.52 micrograms per liter. Some of the first takeaways here, there's interannual variability and spatial variability. It seems that the second season that we've analyzed so far is a stronger bloom year, more bloom activity, and it seems to affect actually throughout the entire lake, which is interesting. If we take some of our preliminary data in regard to comparing in situ tracking, which is the secondary y-axis on all of these graphs, versus grab sampling data on the left-hand side, and again on the map you see where we could do those comparisons, you can see that for each of these stations we see slightly different patterns. So the symbols go to the primary axis, the columns are the levels from in situ tracking, the cumulative signals. And you can see when the symbols are empty, we did not find microcystin, but we still see it in the in situ tracking method for that period in time. Some of the takeaways here that warrant further investigation is there certain areas where the toxin tends to accumulate or where we actually have the blooms originating. So if you look at these two stations, 19E and 19P, you can see that there's an accumulation of the toxins based on in situ tracking that is not reflected in the grab sampling effort and not seen at the other two stations. So if that's the case, is there also a higher potential in those areas for food web accumulation over time? So many more questions that we're looking into. We're looking forward to a third season of observations. We will start to add another component where we do molecular analysis to get at cyanobacterial species composition. This is a resolution that is needed if you want to actually get at which toxin-producing species are present. You can't determine that through microscopy. You actually need to look at molecular signals. And we will have that information to look at population connectivity throughout time and throughout the lake and relate that to the toxin dynamics, as well as the environmental conditions, both physical, chemical, and hydrological conditions. To take a step back, looking at the Falls Lake nutrient study as a whole, we do believe that our information can guide some of the assessment strategies, both in regard to which toxins are relevant, and also what's the baseline on toxin producing species that might lead to molecular methodologies that can allow you to detect them early on within the system. We will also get some more information back about potential hotspots for both the species as well as the toxins. And we are confident that our study can inform nutrient management approaches and also be relevant for statistical modeling and prediction to assess the risk to ecosystem health, the drinking water supply, and recreational uses in the immediate future. And with that, I want to thank the Department of Environmental Quality for all their collaborative support. And of course, my Plankton Ecology Lab, my students, and my technician for all their hard work. And thank you for your attention and time. And I'm looking forward to some of your questions.